Hi, I'm Sean at Food Claims. At Food Claims Scientific Services, food testing and laboratory analysis is conducted at one of 150 laboratories in 37 countries. Food Claims specialise in protecting small businesses and medium-sized enterprises within the food and drinks industry with a complete range of food testing and laboratory analysis to ensure safety, quality and the legality of manufacturing operations. We provide cost-effective and robust laboratory analysis systems for our clients. Our food testing services deliver high-quality UKAS accredited laboratory analysis to demanding deadlines and budgets to ensure Food Standards Agency compliance and UKAS accredited certification. Why pay more? Protect your business from a food claim and your bottom line.
Okay, hello. I'm going to talk about identification, particularly bacterial identification, although some of the principles I cover will apply to other organisms, microorganisms other than bacteria, but primarily bacterial identification, or ID for short. What's identification? Well, identification is when you look at the characteristics of an unknown strain, an unknown isolate, and compare those characteristics to those of known bacteria in this case. And usually, for a full identification, we want the species name, which is 2 words. It's a binomial system, as you should know. So we want to say that this particular unknown is Escherichia coli, or E. coli for short, or it's Staphylococcus aureus, or whatever. So that's the principle of identification. But what's a species? Well, in microbiology, a species is defined generally by the characteristics of a particular organism. So all the members of a species will share a lot of characteristics. All their test results will be pretty much the same. Unfortunately, from the point of ID, a lot of strains or variants within a species have slightly different characteristics. So that obviously complicates the issue. Not every member of a particular species will have exactly the same characteristics and give exactly the same test results. I've also mentioned that word strain. Well, what's a strain? Well, a strain is normally a microbial culture, and it's the result of a single isolation of a particular organism. So every time you isolate a particular microbe, a particular bacterium, it's a different strain. It's very important to not mix the word strain up with species. So the way I often tell students to try and remember it is that there's only one species of E. coli, Escherichia coli, which is clearly defined by its characteristics. How many strains of E. coli are there? There's an indefinite number, because every time you isolate it, it becomes a different strain. So you can isolate it 50,000 times, and that's 50,000 strains. They're all the same species, E. coli. So don't ever mix up the word species and strain. Now, if you've got a particular bacterium, you might identify it. You get a full ID, which means you get the species name. A partial ID might be that you've only got the genus or the family, which is pretty useless, actually. The family is far too big a group, far too vague, and we're going to be looking at one particular family in this podcast later. A genus sometimes is okay, but really a full ID means the species. And then even when we've got the species, we sometimes want to do extra tests to what's called type within that species. That's particularly important in epidemiology and cross-infection studies, that we might want to say, well, it isn't just E. coli, but it's a particular type of E. coli, like O157, which you may have heard of. It's a particularly nasty variant of E. coli. It's a serotype. And also, if you want to trace an infection within a hospital-acquired infections, then again typing might be required. I'm not covering that typing now. We're going to concentrate mainly on identification, particularly bacterial identification. Now, what tests do we use for bacterial ID? Well, we can look at the morphology of the colonies. We grow up the bacteria usually on agar plates. Got a typical plate here, and we can look at the types of colony that it's formed. And assuming that it's all one type of colony, which means it's a pure culture, then we might better hazard some sort of guess about its ID. But colonial morphology is rather limited. An awful lot of bacteria on agar plates like this look exactly the same, so it's rather difficult. We could do a gram stain, okay, very common stain in bacteriology. And again, we can get some information that will help us in the identification. So we can look at the shape of the cells, are they rods or cocci? we can look at their aggregations. Are they in chains or bunches and things like that? But that's about it, okay? And are they gram-positive or gram-negative? So we're quite limited. We can look at growth characteristics of a bacteria and what temperature it grows at. And more particularly for ID, what's quite useful is the atmosphere, what gaseous requirements it has. So some bacteria must have oxygen, they're strict aerobes. Some can grow with or without oxygen, they're what's called facultative, and some can grow only anaerobically. So that's quite a useful test. What we want from an identification test is something that's reliable, consistent, not too difficult to do, and also isn't too expensive. So we want reproducibility above all. And something that also discriminates. Tests should be able to discriminate between one type of bacterium and another. For instance, if you had a test and all the bacteria gave the same result, it would be a useless test and you'd scrap it. You wouldn't use it. So we can use growth. There's also some miscellaneous tests that we use a lot in bacteriology. One is motility. We look at whether microbes can move in a hanging drop, and that's really usually demonstrates the presence of flagella, or one flagellum or several. And smell. Actually, surprisingly enough, some bacteria have very distinctive smells, but again, a lot have very similar smells. They just smell unpleasant sometimes, and that's not terribly useful. So I'm now going to move on to the most important group in terms of number for testing and doing ID, and that's biochemical tests. All sorts of biochemical tests, breakdowns of substrates, testing to see whether a particular product is formed, testing to see whether the bacteria can use a particular substrate for a particular purpose, for carbon or nitrogen and stuff like that. And there are literally hundreds of biochemical tests. And what does the lab do? Does it want to do all of them? For accuracy in ID, the more tests you do, the better. But you've also got to compromise a bit on that because you've got time, you want the results quickly, and there's also expense. So you don't want to do more tests than are absolutely necessary. One of the things a lot of labs will use, very commonly, are commercial test kits. And we're going to look at one now in detail, and that's called the API 20E system. 20, because it's got 20 tests in it, and E, because it's only used for Enterobacteriaceae and a few related organisms. Enterobacteriaceae are gram-negative rods. They're commonly found in the gut. They are found in other places as well. So we can use a 20E strip if we already have a fairly good idea that we've got an Enterobacteriaceae. If you've got some other type of bacterium, then you'd be wasting your money and your time setting up the strip. The company that makes the API 20E is called, its API stands for Analytical Profile Index, loosely translated from the French. It's a French company called bioMérieux. They also make other strips as well. We'll look at some of the other types of strip they make as well. And of course there's rival companies that have similar systems. And what the API 20E has is it has 20 biochemical tests. They're all biochemical tests in a very convenient plastic strip.